Hello, my name is Dr. Roderick L. Roll, and today I'll be talking about antibiotics and how they control bacteria by altering or affecting their cell wall. This is another series of drugs that are inhibiting bacterial or microbial growth, and I'm going to cover many of these drugs that are in this slide image. Before we cover how the drug can affect the wall, let's look at the actual wall. The wall is composed of a sugar backbone, NAG versus NAM, NAG and acetylglucosamine, NAM and acetylmuramic acid. Those are the two modified sugars. They're modified versions of glucose. Notice how each of the layers is connected to another layer via a tetrapeptide and a pentapeptide. The tetrapeptide is outlined with a red circle, and it's four amino acids. And the pentapeptide is outlined by a yellow circle, and it is five amino acids. Both of these tetra and pentapeptide holds layer one of NAGNAM to layer two of NAGNAM to layer three of NAGNAM, and so on. Here's another way to look at it. Layer one NAGNAM is attached to layer 2 through those two segments of amino acids that are linked together, the tetrapeptide peptide or the pentapeptide. How do antibiotics affect the cell wall of bacteria? So that's what we'll dive deep into now. We will first talk about beta lactams. Beta lactams are a category of drug that have functional groups that contain a beta-lactam ring. Notice in the picture, I'm pointing to a beta-lactam ring. So whenever you see the molecular structure of a drug and it has this shape, that is a beta-lactam ring, and it will be a drug category that's effective against bacteria. In this slide, we're looking at several examples of beta-lactams and chemically altered versions of them. So the natural beta-lactam is penicillin G, and the others in the picture are modified versions, such as penicillin, the one that's called methicillin, that is a semi-synthetic. So they chemically altered the original penicillin to make it stronger, to make it last longer, to make it more potent. Another semi-synthetic version is this cephalo uh, cephalothrin, and then the last one. So all of those are examples. Monobacterium, cephalosporin, and penicillin are the three categories of beta-lactams. Natural penicillin and other natural beta-lactams affect gram-positive bacteria and spirochetes, such as Treponema pallidum, the causative agent of syphilis. So notice they will affect gram-positives, the ones that have that very thick peptidoglycan wall, but they will not affect the one to the right that is called a gram-negative. Why? The gram-negative have an outer membrane that will not allow that beta-lactam to even gain access to the wall to destroy the wall. The only way around that inhibition is to give another drug, or if penicillin was somehow able to cross that outer membrane and get to the wall, then it could destroy it. So therefore, a hole like a porin would have to allow penicillin to enter to destroy that gram-negative bacteria. Now, if we alter the beta-lactam, it is going to increase its killing capability. It'll make it have a broader killing range than the normal beta-lactam. So let's look at the wall and how the wall is synthesized. The bacteria is going to produce these bricks. These bricks are the N-acetylglucosamine and the N-acetylmuramic acid, or NAG and NAM. So from now on, I'll just call it NAG versus NAM. Look at this depiction, and you can see the NAG and the NAM, and they're indicated by either a red hexagon or a pink hexagon. The pink one is NAM, and the red one is NAG. A pentapeptide, which is 5 amino acids, and the actual amino acid is glycine. So 5 glycines linked together are going to form this cross linkage. This is going to hold the wall together. If you could somehow inhibit that glycine, 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 glycine link, then now the layers float away. That is how the beta lactams will work. Beta lactams will inhibit linkage between NAM and NAM. So the pentapeptide, those glycines, are going to be between the pink uh, hexagonal shaped structures. So when the patient takes beta lactam, it inhibits the formation of the pentapeptide. Therefore, the layers float away. The peptidoglycan layer will form, but it will not be able to be anchored to the existing layer. Bacteria will have a weakened cell wall and eventually lyse. In order for the bacteria to reproduce, it must produce and insert 2 million new NAGNAMs. So notice this bacteria is able to produce it, but cannot insert it into its cell wall. This means the bacteria will rupture, will be destroyed. So not exposed to beta-lactam is the image of the bacteria on the left, and the one on the right is when the bacteria is exposed to this beta-lactam. Prevention of cell wall synthesis by inhibiting this pentapeptide from linking adjacent layers is the first category, and that was achieved through the consumption of beta lactams. More common antibiotics prevent cross linkage of NAM and NAM subunit. So we just looked at if we inhibit where that yellow circle is. If we can inhibit that from forming, we stop the layers from being able to be attached to the adjacent layer. Again, you produce the NAG and NAM, but they cannot insert to the previous layer. So no NAM-NAM. So no NAM-NAM, the bacteria cannot reproduce. And no NAM-NAM means you can stop it by altering or inhibiting the red, what a red circle is, that is the tetrapeptide, or by inhibiting where the yellow circle is, which is the pentapeptide. So either way, the layer will not be able to attach. So let's move to another category of drugs. So another group is called vancomycin. This drug binds to the terminus of the tetrapeptide called D-alanine. So look at the picture below. Now we've already talked about the peptide of five glycines. Now let's look at the tetrapeptide. The tetrapeptide is composed of alanine, glycine, lysine, and alanine. Notice the alanine, there's two versions, the right-hand version and the left-hand version. So we call that L-alanine and D-alanine. The D-alanine is the amino acid in question. Vancomycin is going to put a force field around D-alanine. If it can if it can put a force field around D-alanine, then that amino acid cannot attach to the pentapeptide. If it can't attach to that pentapeptide, you cannot attach to layers. Layer 1 cannot attach to layer 2, and layer 2 cannot attach to layer 3. So this is how vancomycin works. So that was our second group. Our second group inhibits layer 1 from attaching to layer 2, but it affects the tetrapeptide. The next category is cycloserine. Cycloserine competitively inhibits two cytoplasmic enzymes that link NAM to NAM. One enzyme converts L-alanine to D-alanine, while the other incorporates D-alanine into the tetrapeptide. So again, let's look at that tetrapeptide, look at that D-alanine that I'm pointing to in the picture. If I can somehow inhibit that D-alanine, put a force field around it, or not even allow it to be produced. If either one of those occur by consuming cycloserine, I again stop the layers from forming. So the third one, cycloserine, inhibits the tetrapeptide from being able to form appropriately or completely. The next category of drug is bacitracin. Bacitracin is a highly toxic drug that must be applied topically. You cannot consume this. This drug will prevent the secretion of NAG and NAM. So the other ones, you secreted, but you were not able to insert the layers to layers. This drug will not even allow you to secrete it. So that is all of the categories that inhibit the wall. The last one, bacitracin, no secretion of NAG and NAM. The next category of drug is isoniazid and ethambutol. Those two will destroy the causative agent of tuberculosis, which is Mycobacterium. Remember, Mycobacterium has a very thick, waxy coat that surrounds its peptidoglycan wall. A lot of drugs are not able to penetrate, and chemicals cannot penetrate that thick, waxy coat. Sonazide and ethambutol will be able to disrupt that. However, since the bacteria reproduces very slow, remember E. coli replicates in 20 minutes. Since this bacteria replicates every 12 to 24 hours, the patient will be on this medication for months to even years if they have tuberculosis. How are some bacteria resistant to these processes? Some bacteria are resistant to certain drugs because they produce enzymes that can deactivate those drugs. We talked about beta-lactam and the natural form being penicillin. That penicillin is going to inhibit the pentapeptide. Therefore, no NAM and NAM can link. But if the bacteria produces penicillinase, and penicillinase is like scissors that cut up penicillin, then penicillin is rendered inactive. So that is how some bacteria can resist these drugs. They have enzymes that can cut up the actual drug, making it inactive. When treating penicillin-resistant staphylococcus, you must use a semi-synthetic penicillin called methicillin. And even some strands may be resistant to methicillin. If a strand becomes resistant to methicillin, it is called methicillin-resistant Staphylococcus aureus, or shortened as MRSA.